The brain is waiting for us to learn about it. Welcome! The topic of today's lecture is sensory systems. We're starting to talk about senses. In this lecture, we'll introduce senses as a whole, and then we'll talk about some of the minor senses. Minor. I kind of don't want to spoil which senses this will be, because my first question will actually be, let's list all the senses that you know. But I'll probably say that we will spend 2 whole weeks on hearing, and we'll spend 2 whole weeks on vision. So today we're talking about everything else. And let's start with answering these basic questions. What senses do we even have? What sensory modalities do exist? And there are probably 3 parts to this question. First, what are the classic senses? Because you probably heard about 5 senses, because the 6th sense is supposed to be something weird, right? These are called Aristotelian senses, after Aristotle, who just happened to be the person who decided to enumerate the senses, that he came up with this random number of 5. So there are these 5 Aristotelian senses. Let's start with naming them. And the second part of the question, think of whether we have any other senses, like not this weird 6th sense from mystery books, but was Aristotle actually right? Is there anything else in us, in our bodies, anything that he overlooked? And the final part of the question, think of other animals, not us, not humans, but all other animals you know. Are there any other senses that other animals have that perhaps are not present in a human? As usual, pause the video, because this is a big question. You will probably need paper and a pencil. Pause the video, write down your answers, and then continue and compare your answers to mine. Try to list as many senses as possible, because it can be a quite long list, to be honest. Alright, what's on your list? Let's start kind of according to the program that I outlined. Let's start with the Aristotelian ones. Aristotle came up with 5 senses: vision, hearing, smell, taste, and touch. But if you thought carefully enough, you would notice that something is iffy with it. So vision and hearing are okay. Smell and taste, sure. What about touch, for example? You know, touch is actually a more complex thing, right? Because pressure and cold are kind of combined into touch, basically because Aristotle called it that way, because he came up with these 5 names. If you think of it, cold and pressure don't feel anything alike, except that both are sensed by skin, largely, although not even always. So let's disassemble touch in several components. At the very least, we can think about pressure, which is like constant pressure, vibration, which is like quick changes in pressure, cold, and heat. And cold and heat, you may think about that as one sense, because it basically reports to you the temperature of the object, but we will see that they actually are reported by different receptors. Then there is pain, and Aristotle probably, I'm not sure what he actually did. Did he include it in touch? Perhaps, but of course pain is weirder. And we talked about pain before. It doesn't always originate in the skin. Our muscles and even our viscera, our internal organs, may be the source of pain. And then there is the vestibular sense. And vestibular sense is obviously not one of those 5, because what is it? It is the sense of balance. Probably the best way to notice that the vestibular sense even exists is to meet people in whom it's disrupted, or maybe to get yourself in a situation in which it is disrupted. But it is a separate sense. The organ for it is located in our head, in the inner ear, and it would basically tells you whether you are standing or lying, for example. The posture of the body is pretty much the same, but the orientation is different. That's the sense that tells you where the up and down is. But then for the posture itself, we can also tell whether we are sitting, for example, or standing. We can tell even with our eyes shut whether our fingers are extended or whether they curled in fists. How do we know that? Obviously, touch is a little bit involved in case of fingers, but how do we know whether our elbow is bent or extended? That's because we have other types of sensors that are located inside our body and that communicate to our brain the position of our joints, muscles, and tendons. And together, these are called proprioREceptors, and we'll talk about them today. And at this point, we mostly exhausted humans, but obviously humans are not the only animals on this planet. And if you look at other animals, it's ridiculous just how diverse and rich the sensory world of some animals is. Vision, of course humans have vision, and human vision is actually pretty good, as we will talk about later. But there are animals who can see things that we cannot. For example, there are many animals who can see ultraviolet. that we called ultraviolet precisely because it's beyond violet and we cannot see it. Some animals can see light polarization. Some animals can see light polarization, and I cannot even explain to you what light polarization is because I've never perceived it and neither did you. I can try to explain it from the physics point of view, but it's just not something that we are aware of. That's not something that's part of our sensory experience. While bees, for example, and birds can see light polarization. Then there is infrared sensation, which is like seeing in infrared. It's like seeing the heat that the prey emits as you're hunting. And snakes have it, but it isn't actually mediated by the eyes. So it is like vision, but without eyes. They have a special organ for that. Then there are variations of hearing that are very much not like human hearing, or at least typical human hearing. Many animals can hear ultrasound. Even rats and mice can hear ultrasound, but then some animals use ultrasound for echolocation. Like bats and dolphins, they emit ultrasound and catch the wave as it reflects from objects and obstacles and potentially prey, and in this way figure out what's around them, even in complete darkness. Isn't it nice? And then there is 3D hearing, which is how you can hear if you listen to how the ground trembles. Then there are several types of touch that we don't have. There is lateral line in fish that we briefly talked about when we discussed escape responses in fish. Then many animals have vibrissae, which are these long hairs that grow on their snout. If you've ever seen a cat, you know those whiskers that they have, which are not like fur, not like human whiskers, but long single hairs sticking out of their face. What are these? Well, it turns out this is a special organ for touch, just really weird touch, and we'll talk briefly about that today. And some animals use these whisker-like structures to feel underwater, like manatees, for example, who don't have a lateral line because they're mammals, they're not fish, but in the process of adapting to the aquatic environment, they figured out how to use their modified hairs to feel the movement of water in the same way in which fish use their lateral line. And then there are electrosensation and magnetosensation, which are again super weird to humans because we don't have it, but it turns out that many aquatic animals have electrosensation. And birds, for example, have magnetosensation. They can directly feel magnetic fields, which is hard for us to imagine. Okay, good. We'll talk about some of these topics later. Now, next question. Coding. What is coding? Basically, the question is about how is information transmitted in the brain from one part of the brain to another, or from a sensory receptor to the brain. Because the natural question is, okay, once the light is caught by the eye, or once the sound is caught by the ear, how is it encoded by neurons? Neurons can spike. How can you encode a perception of something with spikes? And it is, of course, a very complex question, a very complicated one. So let's start with the most run-down version of it. Imagine that there are two parts in the brain, nucleus A and nucleus B. Or maybe you have a sensory organ, and that's the point A, and the point B is the brain. But either way, one part of the nervous system tries to communicate a message to the second part of the nervous system. And in order to have it more accessible, let's consider the simplest scenario possible. We have communication from point A to point B. We have a nerve running from A to B that has only 3 axons. Why 3? Because it's a small number. Let's imagine there are 3 axons. And let's also imagine that the information we need to transmit is fairly simple. It's just the presence or absence and maybe the degree of a certain stimulus. Say it's either nothing, or a little bit of light, or medium amount of light, or very bright light. Or maybe a weak pressure, medium pressure, strong pressure. Or maybe a small object, a medium-sized object, and a large object. Or warm to hotter to really hot. Let's imagine that we communicate where the stimulus is present and the degree, the strength of the stimulus. And also think about what axons can do, what neurons can actually do, what messages they can send. Because neurons cannot communicate complex messages, can they? They don't have that many words in their vocabulary. What can one neuron do? It can spike, or it cannot spike. Your vocabulary consists of one word and silence. And somehow with this very limited vocabulary, you need to communicate this simplistic but still relatively realistic message. How to do that? There are at least 6 possible answers. 6. There are at least 6 possible answers to this question, but some of them are pretty fancy. Pause the video. Take a piece of paper and try to figure out what options do we even have. If you were Archangel Gabriel and the Lord asked you to design the brain from scratch, if you had to make sure that point A communicates to point B and sends this kind of a message, how would you have approached that? Pause the video, prepare some sketches, and then compare your answer to my answer. Done? Okay, let's look at what we have. This slide is rather complicated. We will spend some time here. So let's look at the general layout. There are 3 columns here that correspond to these 3 degrees of stimulus strength. It doesn't have to be 3. You can imagine everything in between as well. But the stimulus can be weak, which I represent with a tiny circle, and it can be medium strength or medium brightness or medium intensity of a smell or something like that, medium loudness, or it can be really big, bright, loud, intense. So from nothing, we go all the way to a lot. Each of these lines is the activity of one neuron. And every time we have a spike, I draw a little line across this line. No spiking, and then there is spiking, and then no spiking again, and then there are 2 spikes. That's basically how it is represented here. You can imagine a timeline running in this direction. The first option, and probably the most simple one, and I bet you came up with this answer, we can just spike the different amount of spikes. So maybe when the stimulus is weak, we spike a little bit. And when the stimulus is stronger or larger or brighter, we spike more. And then for really powerful stimuli, we spike really a lot. This is a nice approach, and it's actually used by the brain all the time. It's called the rate code. The rate of spiking encodes the strength of the message. And that's by far the most common type of communication in the brain, or at least it seems so. A similar but slightly different concept is the population rate code that I call a population code here. It's similar in the sense that the more intense is the stimulus, the stronger it is, the more spikes we have. However, in this case, it's not about changing the frequency of spikes in one fiber, in one axon, but about recruiting, involving in our response more and more different fibers. So maybe when there is no stimulus, there is no spiking, but then if the stimulus is weak, only some axons spike. As it becomes stronger, more axons gets involved until for the strongest stimulus, all of the axons in the system are spiking. And the total effect at the point B, where this information goes, is about the same. You just look, listen, in electrical sense using your synapses to the total amount of spiking that arrives. The difference is that in population coding, in order to decode the message, in order to figure out what message is communicated, it's not enough to listen to one axon. You really have to listen to the entire population. Every one axon communicates only part of a message, and only the population as a whole encodes the whole message. That's a big difference. For rate coding, if every axon follows this logic, listening to any single axon would be enough and would tell you exactly what's going on. Next option, labeled line. We can make every axon communicate a different degree of the stimulus, a different type of the stimulus. Maybe if you see a tiny object, one axon spikes. And when you have a medium-sized object, this first axon shuts down and the second axon spikes. And when you have a large object, the third axon spikes. It's called labeled line. It's a metaphor for those really old school mountain resorts and hotels before electricity was really invented, when every room had a little rope running from this room downstairs to where the hotel employees were sitting, and every rope was tied to a bell, and under every bell was a label. Room number 1, room number 5, room number 26. If you needed something in your hotel room, you would pull on the wire, and somewhere down there, this little bell would ring, and the person would look at the ringing bell and look at the label near the bell and understand which room needs service. That's kind of the same idea here. You have different fibers, and different fibers communicate different messages. Now, which of these so far is simpler? It depends on the situation, right? Which one is easier to encode? Probably rate code. Maybe you detect light. The stronger is the light, the stronger is the stimulation of the fiber. As we had seen in one of the previous lectures, the more you try to depolarize the neuron, the more spikes it produces. And is it easy to decode it? Is it easy to listen on the receiving end? Kind of, yeah. You just connect it to a synapse, basically. The more you activate the synapse, the more neurotransmitter is released. The more neurotransmitter is released, the stronger the postsynaptic neuron is activated. So this is fairly simple. The second one is also simple. At the listening end, literally nothing changes. You just look at the total number of spikes that arrives, and for that you just need a synapse. And at the encoding end, it's also not that complicated, because you can have fibers with different thresholds, different recruitment thresholds. So maybe some fibers are really eager to spike, and they're the first to spike as the stimulus arrives. But then other fibers are a little bit tough, but as the stimulus becomes stronger, they are also recruited. And then some fibers have a really high threshold, so you have to have really intense stimulation for them to get recruited. But the end result is that you communicate the message. What about the labeled line? It's pretty hard to encode, right? It actually requires some thinking, some calculation. But at the receiving end, it's really nice to have a labeled line coming in, because you can just literally wire different neurons to different outcomes. Maybe if you see something tiny, you eat it, but when you see something large, you escape from it. A labeled line would be nice in this case. Now, the next question, can we somehow use not how many spikes were there, but the timing of the spikes? Can we communicate information by the timing? And it turns out that we can, and there are at least two ways of doing that. Let's start with the simpler one, the one with one fiber. So if you have three fibers, all three can be doing that. Let's look at this example. We can encode information by creating bursts. The total number of spikes may be the same, just here they would all be placed at equal intervals, and here in the extreme case they will be communicated by short bursts. So instead of doing pam 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 pam, we'll be doing pam 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 pam, pause, pam 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 pam, pause, and then everything in between. Is it easy to encode? Well, maybe. You just need a special neuron. Is it simple to decode? That's a little bit more tricky, but you can actually build a decoder. It's kind of an advanced topic, so I don't want to go into it, but if you think really deep about how synapses work, perhaps you can figure out how to make a synapse that responds weakly to something like that, when you have 4 spikes at equal distances, but responds strongly to something like that, where you have 2 spikes that are close to each other and then a pause, and then again 2 spikes that are close to each other and then a pause. Can we do population temporal code? Yes, we can, and it is called phase code. And you can see from the picture what it means. Different fibers may spike either in synchrony or out of sync. On this picture, the number of spikes generated by each fiber is again the same for weaker stimuli and for stronger stimuli. But in this case, they are all in synchrony, and in this case, they are all out of sync. Is it easy to decode? Is it easy to encode? It depends, but at least let's be aware that it exists. And I totally forgot to mention this first line, the graded potential. Graded potential almost doesn't belong here, because all the other options use spikes. But the thing is, if your neuron is really tiny, if points A and B are really close to each other, then maybe spikes are not even necessary. If you only have to travel a few microns, you can afford to just depolarize the very cell as a whole. You can have a non-spiking neuron. And indeed, in some cases in our brain, when distances for the signal to travel are very short, some neurons just don't even bother to spike, because then you can just change the potential of the entire cell. And the stronger the stimulus, the higher the potential becomes. We will talk about that when we discuss vision, because that's how some of the cells encode information in our retinas, in the eye. But that's one of the few places where you have this situation. For all other systems in the brain, you cannot afford that. These terms and these ideas are worth remembering, because we will come back to them again and again as we discuss different sensory systems. Now let's talk about our first sense, about touch. Let's start with touch receptors. Now this figure is really overloaded, too detailed for this course. Basically, I want you to concentrate on one message here. There are several types of touch receptors. Let's look. There are free nerves endings that are about pain, but then also there are Meissner's corpuscles, and then there are Pacinian corpuscles, and then there are Ruffini's endings. All of these are sensory neurons, and all of them have different shape, and all of them communicate to the brain different aspects of what we call touch. Different receptors communicate constant pressure and vibration, What does it mean? If you press on something or if you have a heavy object in your hand, you feel this pressure and it's communicated by one type of neural endings in your skin. And then there is vibration. When something touches your skin lightly, and different type of receptors will detect that. And what happens in our brain is that it synthesizes all these signals into one perception. If you've ever played these little games where you need to put your hand in a bag and there are different toys there and you need to figure out what is this that you're touching, it's kind of fun and it's kind of hard, but we learn how to do that. And what's interesting is that there are all these types of nerves with names of people who first studied them that communicate different types of information. And then our brain synthesizes that into one tactile reality. And let's concentrate on one aspect, which is cold and warm receptors, because here we can illustrate several interesting points. These are two types of receptors. Again, both communicate to the brain, and they have a certain sensitivity curves that you can build and look at them. They're shown on this plot. And here, for cold receptors and for warm receptors, you can see how they respond to a change in temperature. Here we have a regional temperature of 38 degrees, which is slightly warmer than the surface of our skin. And then we have a step to 32 degrees, which is colder than the normal temperature of our skin, and it returns back. And you can see how cold receptor starts spiking more, while the warm receptor does the opposite. From relatively strong spiking, it goes to no spiking. There are at least three learning points that we can have here. One is that when something changes, receptors spike more, and then they accommodate. And this is a very common theme for touch, for cold, for smell. The change in something is more noticeable than the stable state. The change in pressure is more noticeable than a constant pressure. The change in brightness is more noticeable than constant brightness. So here we have an interesting situation when a very useful type of information processing is also relatively simple and can actually be performed by a single neural cell. That was learning point number one, accommodation. Learning point number two, the sensitivity of the cold receptor and of the warm receptor actually overlap. There is this intersection region where both of them are activated. And that's interesting because it again shows kind of how our brain creates reality from all these pieces of evidence that arrive from different receptors. If you want to figure the temperature of the object that you're touching, and you have some contribution from cold receptors and some contribution from warm receptors, you can just look into how much cold and how much warm signal you have. If you have a lot of warm and little cold, okay, it means you're somewhere around here. If you have only cold, okay, it means that you're somewhere around here. But if you introspect that, you cannot separate these two modalities. It's not something that's available to us consciously. If you try to really concentrate on both sensations, you won't be able to feel cold and warmth separately. We just synthesize that into one experience. Point number three. Sometimes weird things happen. Really hot water would also activate cold receptors. And we actually feel it sometimes when we put our hands under the water stream. Sometimes there is this confusion where we are not even sure whether the water is cold or hot. Sometimes for a split second it feels like we're not sure what's going on. And that's not something that's built in. It's not something that was introduced in our sensory system by evolution. It's just a quirk that's little enough to persist because it doesn't harm us too much, but that at the same time is pretty weird. Okay, next, a question. There are two chemicals that activate cold and warm receptors respectively. Guess what these chemicals are. There is a chemical and you know it. You know this chemical. The receptor for cold is supposed to respond to cold, but there is a chemical that would trick it, and the other way around, there is a chemical that tricks warm receptors. Can you guess them? That's a short question but interesting, so pause the video, think, then continue. Alright, did you figure it out? What's cold? What feels cold even when it's actually warm? And what's hot? What feels hot even when it actually is not? Mint and peppers, or maybe mustard or horseradish. These are slightly different chemicals. Mint activates our cold receptor. The chemical is called menthol, while peppers and the chemical is called capsaicin activate our warm receptors. And we humans are strange. We like experiences so much that we eat both. Mint is not used in Western cuisine that much, but I guess it's used in drinks. But, you know, peppers and garlic and mustard are everywhere. I think British people actually didn't like any spices, and early Americans were pretty bland, but now it had all changed, right? And see also the same theme that all toxins and agonists and antagonists are either in venoms or in plants. In this case, plants don't want to be eaten. So they created these chemicals that just mess with your receptors in the hope that you would not eat them. Little did they know that humans are so weird that they would cultivate them specifically to have access to these chemicals. Well, plants are probably happy about that. Are plants happy? I would be happy if I were a plant. Now, this is a pretty specialized slide, but bear with me, it leads to an interesting consequence. How are these fibers connected? There is a receptor somewhere over here, and this receptor is connected to an axon, and then this axon travels up the spinal cord until it gets to medulla. What is medulla? We haven't mentioned this word before because we hadn't looked at brain morphology, but it's something in the brainstem. So it's basically here. What does it mean in practice? It means that we had just located the longest nerve in our body. It's kind of logical that longest nerves in our body would be either brain talking to your foot or foot talking to your brain. But we had just talked in the previous lecture about motor neurons, that no neurons from the brain actually connect directly to muscles. We have those motor neurons that are sitting in the spinal cord that talk to muscles. So when you move your foot, a neuron from your head comes to your spinal cord at the back, and then a neuron from there goes to the muscle that moves the foot. Both are pretty long because one goes your entire trunk and the other goes from the middle of your back into your foot, but none really traverses your entire body. It turns out that here, for sensory neurons, we don't have this intermediate stop in the spinal cord. So the neuron to sense the world with your great toe communicates all the way to the brainstem. And this is the longest neuron in your body. It goes from your foot to your head, and the cell body is sitting somewhere in between. It also tells us which is the longest neuron in the known universe. It's basically the same neuron, just not in a human. Which is the largest animal? Which is the longest animal that you know? That's a whale, right? And a whale is a vertebrate. Its closest living relative that doesn't live in the water is a hippopotamus. Either way, the general layout of the nervous system in a whale is pretty close to a human, and it's what's shown in this picture, which means that there is a neuron in a whale that communicates from the flipper, from the very end, from the tail, all the way into the head. Can you imagine that? A cell, a single cell that runs all the length of a blue whale, and it takes several seconds for the whale to feel what's going on with its tail. Now, let's briefly talk about how people study somatosensation, touch, tactile receptors. And here's the question for you. If you study touch, if you want to study touch, or any other sense for this matter, would you rather use an animal that's strong in this sense, that relies on it a lot, or the animal that basically doesn't care about it? That's a simple question. You probably have your answer. It kind of doesn't make sense to use an animal that doesn't see in order to study sight. And if we push this logic all the way, it kind of makes sense that to study touch, for example, you would try to find an animal that relies on touch a lot. This principle is called the Krog's principle, and Krog got a Nobel Prize at some point for his work. And the principle is that for any problem in physiology, there is almost certainly an ideal animal to study it. When applied to neuroscience, it's sometimes also called the principle of a champion species. For any type of behavior, for any type of perception, for any type of motor output, you can find an animal that is so good in this behavior, or in which this behavior is so noticeable, or so isolated, or so easy to manipulate, that this animal looks as if it was created, that's the joke, to be used for these studies. So what animals are champions of touch? The best touch ever is probably that in a star-nosed mole. They have these fingers on their nose. There are some lovely videos about them on YouTube. Please watch them. They have something like fingers on their noses, and they move them in order to touch the environment because they live underground and they hunt using these appendages. They basically hunt by touch. They dig the earth, and then they touch what they had just dug, and the moment they touch anything edible, which is in this case typically a worm or an insect, they immediately eat it. This organ has a special name, Eimer's organ, and the concentration of touch receptors in the skin in these organs is bizarre. So when people wanted to study how touch works, that's the animal and that's the organ that they used. But star-nosed moths are not the only species that are famous for its touch, and another animal is used actually not to study touch itself, but kind of to study how brain works in general and how the cortex in mammals operates. But in this case it also uses touch, and the animal is a mouse or a rat, and the touch is not our normal touch. It's not something that we have in our fingers or even in our noses, but rather a thing called whiskers. Whiskers or vibrissae are those long hairs that you can see on the snout of a rat or a mouse or a dog or a cat, and it turns out that they don't just walk with them sticking out. This is helpful to begin with, right? If something is sticking out of your snout and every now and then you touch objects with it, you actually know, you kind of know where these objects are before you bump into them. So it's a really handy tool if you live in a hole underground, because underground is dark and you cannot see anything, and you pretty much cannot hear walls. You can hear reflection of your steps perhaps, but not if the hole is really tiny. But if you have these long whiskers sticking from your snout, you can touch the left wall and the right wall pretty much all the time, so you always know where you are. But moreover, these animals move their whiskers all the time. There is a behavior called whisking, and if you look at their cortex, they have a special part of the cortex that represents information coming from these whiskers, and it looks special with a microscope because each of these whiskers has a representation in this cortex, and it's called the barrel cortex because I guess somebody thought that they resemble barrels. Here each whisker is represented by this barrel, and there are links in the description to some nice videos of these rats. They usually recorded in white background and you have a silhouette, and then you have to record it in high frame rate because the process is really fast. And then if you do that, you notice that no rat and no mouse would just approach an object and be content with smelling it passively or looking at it. It always does this motion with its whiskers, and it studies the object from all the sides. It's like touching the object, except it doesn't use its paws, it uses these whiskers to understand its shape and the material of which it is made, and it can figure that out even in complete darkness. That's just how cool this alternative type of touch is. So, the take-home messages for the touch segment. What is perceived as one sensory reality can actually come from very different receptors, like the case of our touch receptors, and our brain just glues that all together. Even for warmth alone, cold and warm sensations overlap to a large degree, but we synthesize one sensory reality from these disparate inputs, which makes the second point here. The third point, sometimes you have non-specific activation, for example, really warm water activating the cold receptor, or a chemical stimulus activating the receptor. Or another classic example, if something hits your eye, sometimes you see a flash of light. Our eye is not a receptor that was designed to feel fists, but if someone hits you in the eye, you can see these proverbial stars that in all Disney movies are flying around the character. It's an example of a non-specific activation of receptors, and here we have seen it in the case of touch. And finally, the fourth point, champion species. The idea that for every question there probably exists a species in which studying this question would be simpler than in any other species. Next topic, proprioREception. What is proprioREception? ProprioREception is the sense that tells you about the position of your joints and muscles. We already talked about one of proprioREceptors, and that was when we talked about knee-jerk reflex, about the patellar reflex, because there was a special fiber in the muscle that communicates to the spinal cord the length of the muscle. And this type of a receptor, which is actually not a neuron, but a specialized modified muscle cell that doesn't contract, it dutifully talks to a neuron so that the neuron could send this information to the spinal cord. What is the length of the muscle currently? How stretched it is? But that's not the only proprioceptor. Another one is called Golgi tendon organ. It's located in our tendons, and it doesn't communicate the length of the tendon because tendons are pretty robust. They're not supposed to stretch. that much. It communicates the amount of force that stretches the tendon right now. And from these two pieces of information, we basically know the posture of our body. If you close your eyes, if you can, for example, touch your nose with your eyes closed, if you can do it, it means that your proprioceptors work. You know the position of your body without seeing it. What happens when a person has its proprioception damaged? In case of peripheral injury, for example, or some weird autoimmune disease that attacks this particular type of receptors, they cannot touch nose with their eyes closed. And if you think of it, they cannot move reasonably well without seeing the position of their own body. It makes life really hard. It means that with their eyes closed or with lights switched off, they would typically collapse on the floor. If they're lucky, they can still feel their skin. A little bit of information will actually reach their brain, because depending on whether your arm is extended or not, for example, the skin receptors will communicate to the brain a little bit differently. And that's how we figure out the position of our joints. However, it will be really hard for them. Clinging to things is another thing. For example, if you pick an egg, you somehow manage to pick it without dropping it, without letting it drop, or without crashing it, which means that you can measure the amount of strength you use to grip things. And it is not a trivial thing to do, because it means that from the spiking that comes from your pressure receptors, you have an estimation for how strongly you grip. People with proprioception damage cannot do that. So they tend to grip things too strongly, because if they underdo it, things just fall down, which means that they have real problems with things like eggs or tender pastries or other fragile objects. It's hard to work with fragile objects if essentially you live in a world of two options: either barely grip or grip with your entire force. Another thing that becomes problematic, and another thing that we never think of when we think about proprioception, is voice control, which is whether you talk high or whether you talk low, and ability to sing because of that. We can do that even if we're talking on a subway in a really noisy environment. Even if we don't hear ourselves, we can still modulate our voice. It turns out that without proprioception, we don't have this information from our vocal cords about the amount of stretch that is put in the muscles that stretch our vocal cords. And we will talk about the vocal cords later when we talk about hearing. If you lose your proprioception suddenly, you can only use your ears to figure out the sound that you produce. And then the last one, which is perhaps the most hard to imagine if your proprioception works properly, is that they don't have a feeling of their body if they don't move. Because when you move, you always touch things. At least the clothes that you wear, they always press slightly differently on your skin when you move. But when you don't move, the only information from your body about the position of it comes from your proprioceptors, which means that without this feeling, your body kind of stops existing the moment that you stop moving. And one example of a person with this condition is described in the book of Oliver Sacks, The Man Who Mistook His Wife for a Hat, which, by the way, is the first book anybody interested in the brain needs to read. If you're interested in neuroscience, this is the book number 0 on your reading list. It's even before book number 1. The story is called The Disembodied Lady. Interestingly, alcohol screws up proprioception, which explains some of the phenomena that strongly intoxicated people experience. Now I will describe a little lab. It's called 2-point discrimination, and it is a nice and quick way to figure out which parts of your body are more sensitive to touch and which parts are not. We will look at a very specific thing called 2-point discrimination, which is just how fine our representation of different parts of our body is. What does it mean? Imagine that you touch something and there are 2 points there, 2 corners of an object or 2 tiny objects that are relatively close to each other. Just how close they have to be for your brain to perceive it as one stimulus, as one object. If there are 2 grains of rice, how close they have to be in order for your body to communicate it to your brain as one sensation and not two different sensations that you can both feel. And one way people do that at school, for example, is by having these special plastic devices that have little pins at different distances from each other, and so you can touch your skin at different parts of your body and kind of feel whether it feels like one touch or two touches. A better way to do that, of course, would be with one person and another person, one person touching, another person sitting with their eyes closed and trying to figure out whether they experience one touch or two, because in this case, the person who are being tested doesn't know what's actually happening. And then the person who is stimulating them, they can alternate unpredictably between one touch and two touches and try to make their partner to guess. And if you note all the results, you can figure out at which point they can no longer feel whether it was one touch or two. But in a quick and dirty scenario, you can do it on yourself. And if you don't have this special thing, we can kind of make it on our own. And at this point, I'll probably grow so that you could see my fingers better. Here you are. Let's say we have a ruler and something like scissors. So what can you do? You can open the scissors a little bit and use the ruler to figure out how far the two corners are. And then you can touch this part, or like, let's hold them like that. Touch this part or touch this part. We can compare, for example, fingertips and the palm of our hand and the back of our hand and maybe the forearm or something like that. If you feel adventurous, you can go like to your leg or to your back. In a classroom, it's inappropriate, but if you're at home, you can totally do that. If you don't have scissors, you can use a pin. And the way you do it with a pin, you basically unbend it. And then you have these little things, and you can bring them closer or you can bring them further away. And you can use a ruler and figure out with a ruler how close they are. And then if you're creative with your bending techniques, you can have all sorts of distances between the two. And then your goal is basically to sit with your eyes closed and to press it to your skin and to let it go further and further away until it starts to feel like two different touches and not one touch. And then once it happened and it no longer feels like one object but more like two distinct objects, then you immediately open your eyes and take the ruler and measure how far they were from each other. And then you repeat it again and again, and you compare the sensitivity of your fingertips to the sensitivity of your palm, to that on the back of your hand, and to that somewhere on your hand. And you can add other parts of your body if you wish to. So what's your guess? What part of your body has most sensitive skin in this sense? Not in terms of detecting the lightest touch, but having lots of information about the structure of this touch sensation. In terms of 2 points discrimination. I guess that your guess is close to my guess, which is fingertips are probably the most sensitive because we use it to sense everything around us. And then probably some part of the hand it will be less sensitive, and then the further you go, the less sensitive it will be. If you want real competition, you want your forearm to compete with your leg or with your foot, or your neck to compete with your forearm, or the palm to compete with the back of your hand. These are not trivial questions, but the fact that the fingertips are more sensitive than your bum, is probably clear. So then the next question, what do you think? Which part has more representation in the brain? If you look at different parts of the brain, and some parts of the brain are processing information from the fingers, and some parts of the brain are processing information from your back or from your forearm. Tactile information, information about touch. Which part will be larger, fingertips or the back? Probably fingertips, right? And if you look into your brain, that's what you see. So how to read this image? This is a cross-section of the cortex. And you can see that the face is actually represented by the large area of the cortex. Look at that. This entire part is the face. And the hand, including the fingers, is represented by a large area in the cortex. This entire part is the hand. While legs and the back, for example, they barely have any representation compared to the actual area of the skin. our back is not particularly sensitive. It can sense touch, but it cannot figure out what sort of touch is it, unlike our fingers. And our face, of course, because, all the food comes here. We use our faces to explore the world in a way. So face is an important area as well. Okay, so 2 questions here. First, why information from the fingers would be processed by a larger area of the brain? And I guess the answer is fairly obvious, right? If we get more information with your fingers, then just the sheer volume information is larger, and then you have to have more neurons to analyze this information. It's just more stuff to analyze, so it's only logical that a larger computational power, more hardware, will be dedicated to it. But then the second question is why we can have maps like these, why we even can have a drawing like that, why fingers are close to each other, why aren't they spread all over the brain, why fingers are close to the palm and palm is close to the hand, why the face is represented like something, why is there a face area, why is it not like a lip in one corner and the other lip, the lower lip, in some entirely different part, why is there a map? And this is called a map, by the way. We'll talk about maps in the future. Now, that's an interesting question, so think about that, pause the video, answer, then continue the video and compare it to the answer that I can give. What's your answer? Why related areas are kind of close to each other? Okay, let's think. So if you receive information about touch and you're trying to figure out what is it that you're touching with your hand, for example, like you put your hand out there and you have some sensation and now you're trying to figure out what it means. In this situation, what's more useful for you? To compare the sensation of this finger to the sensation of this finger nearby, to compare what this finger is telling to your brain to this one, or say your finger to the elbow on the other hand, the comparison of which 2 streams of information is more valuable. I guess you agree with me that comparing what these 2 fingers are telling you is more important because they are exploring the world together, while the elbow on the other hand is doing some other stuff. So for any part of your skin, comparison of 2 areas that are close to each other is more meaningful than a comparison of 2 areas that are really far apart. And that means that if you want to save on wiring, if you want your accents to be shorter, you want the areas that represent these 2 parts of skin to be close in the brain. The representations of these 2 areas of your skin, the neurons that work with this information, are better to be located closer to each other in the brain. And if you follow this logic all the way through, you get the idea that the entire hand should be represented kind of in one place, but the neurons that analyze information from different fingers should be located kind of in the same order in which your fingers are located. And then the fingers should be near the part for the palm, which should be near the part for the thumb, which should be near the part for the wrist, and so on. Every now and then you will have a jump, like your forehead and your hands are actually close to each other, but you will have relatively few jumps. In most cases, you'll be able to have this more or less continuous representation of your body. And this makes pictures like that possible, and it makes an even funnier picture of this kind possible, which is called a sensory homunculus. What is a sensory homunculus? A homunculus is a small person, small human-like body. It's Latin, I guess. Alchemists really wanted to make this fake human in a tube. That was the dream of alchemists together with the philosopher's stone. So what you can do, you can draw this tiny human-like creature, but make different parts of this human-like creature larger and smaller, depending on whether they have a larger or smaller representation in the sensory cortex. For example, if hands have a lot of representation, then let's give it large hands. And if the face has a lot of representation, then let's give it large face. And then if legs are small, okay, it will have smaller legs then. And here how it looks like, and pretty funny and memorable. And I guess that's why people like them, because they're memorable. So I went on Google Images and just found all homunculi I could find, including these three-dimensional ones, and they're pretty funny, right? The face and the lips and the tongue especially have really big representations, as do the hands and fingers, and that's the main take-home message that we can have here. An interesting aside, in humans, toes and feet are kind of close to the part that represents genitalia. And that's one of those breaks in representation because these are not immediately close in a real human, but for some reason that's how these parts are represented in the cortex. And I'm not exactly sure about that. But my guess is that it has something to do with feet fetish being the most common sexual fetish among humans, because many people find feet strangely arousing. And my guess is that it may have something to do with how in every person their cortices are innervated with sensory fibers coming from different parts of their body. Because if this innervation in your own body is always like that and there is a little bit of crosstalk, maybe that's how you start feeling about bodies in general. So that's a hypothesis. I'm not sure whether it's true. A quick search on Google Scholar shows that it is an actual hypothesis that people have. I think it's curious. And this concludes our lecture on sensation in general, proprioception, and touch. Thank you for listening and tune in for the next part.
The brain is waiting for us to learn about it.